The first recipient in this panel is Dr. Hongxu Xian of the Versity Blood Research Institute in Milwaukee, and my dinner companion last night. She's receiving a $500,000 grant for her independent research. So we congratulate you and have you have a seat right there. All right. Next we have Dr. Vivek Unni from Columbia University. I'm sorry, he's Oregon Health Science University. It's his, with a $500,000 award. Next up is Dr. Fulin Ma of the University of Pittsburgh, $500,000. And from the University of Texas Southwestern, we have Dr. Laurent Calvier, another $500,000 recipient. Hi there. Good to see you. Let me make sure this is on. Okay. So thank you all for joining us. It's a pleasure to have each of you. I'm so glad to have you in person to collaborate like this and share. Let's start with Hongxu.
Okay.
Tell us a little bit about yourself and what the purpose of your research is. It should be okay if you hold it up close.
How much time I have?
Well, see, what she's referencing is we had dinner last night for 2 hours and 45 minutes.
Yes.
We don't have that much time.
Mike is really a big fan of my story. Really a big fan.
About 10 minutes or so.
Yeah. So hi everyone. I'm Hongxu Xian. Born and grown up in China. I did my undergraduate school in south part of China, Xiamen, a city very south part, and then close to Taiwan. Then 2014, I went to Singapore to start my graduate training with a very nice mentor as my PhD advisor, who encouraged me on this career. So at that time, I really didn't know I'm going to become a scientist or independent investigator at all. I was studying the mitochondria. Probably you learn from some of the textbook. It's an energy powerhouse to provide your energy every day for our daily life. It's pretty much the battery, like Tiffany for the car. Yeah, so I was studying the mitochondria, how it moves inside of each cell. Just it's pure cell biology, very basic research. Know nothing about Alzheimer's disease. Really know nothing about the disease. Then before pandemic, I moved to San Diego. It was a very challenging, new experience to start my postdoc training. Actually, my postdoc mentor was also a CAR-T 2017 awardee, Michael Karen. Yes.
So this is really something else. This is a full circle moment here.
Wow, yeah. And then at that time, before I joined the lab, he was supported by CAR-T in 2017 to study in the brain how the microglia, just now Peter mentioned, how the inside of the microglia, how mitochondria can work. Maybe it has something to do with your C1q protein also, or synaptic loss. We didn't know, but that was 2017. And then I started my position with him 2019 before pandemic, trying to study mitochondria inside of the brain. But somehow it's so many detours, and based on my cell biology training, we actually found my postdoc training study is really about the DNA. So mitochondria evolutionarily, it evolves from bacteria, invade our cells thousands of years ago. So bacteria, like our cell, we have our genomic DNA, we inherited, or we have the ApoE, this gene in the genome DNA. But bacteria also has their own set of the DNA. So it's called, it's mitochondrial DNA, but after bacteria invade our cell, it becomes a part of our cell DNA, it's mitochondrial DNA. So over these years, people always think the mitochondrial DNA has something to do with our energy, our engine, or ATP generation. But it turns out, especially in some innate immune cells, such as microglia, which microglia is the innate cells inside of the brain. But in our bodies, we have so many types of the immune cells. Particularly in these cells, mitochondrial DNA can be damaged, or in a case when you have the disease, you have different infections, our cell can sense this stress, and this DNA immediately get damaged and then released, which has something to do with the DNA damage and the DNA repair machinery. And then this DNA, once it's damaged, it gets released from the mitochondria to smear the whole cell or even out of the cell. And this released DNA can be sensed by different immune cells in our body. So that was my study, how this DNA can trigger the downstream immune cell responses we call as inflammation. Particularly in this card found, or this project, we were trying to study how this information in the neuron, in the brain, has something to do with this DNA. And especially in Alzheimer's disease or just the natural aged people, once you turn to 85 or 90, you will have very high plasma DNA, mitochondrial DNA in your blood. We found that this has nothing to do with the immune cells talk with the innate immune cells like microglia in the brain. So it's like a circle back generated from mitochondria in your cells when your cell try to. We think why initially the DNA gets released is because it's engineered to help the cells to fight against the infectious disease. Like when you have the SARS-CoV-2 infection, mitochondria get damaged, and then this DNA released in a way to to engage the inflammation. Like we have the pain, we have the scratch, we have the fever. It's all types of immune responses, but beneficial to our body to fight against this infection. But some people, after this disease, the virus get cleared out. Some people get back to normal, they get back to the healthy state. But some people that turn out to have the chronic disease, like Alzheimer's disease to me is an autoimmune disease, or it's a chronic autoimmune disorders, because they didn't get this mitochondrial release turned off properly, and this DNA keep being high or sustained in the blood, then it triggers long-term problem. It's like you have the fire alarm at the beginning, the first ring, because you have the fire. It's true. It's a way for you to respond for the fire. But once it's triggered, once this fire is eliminated, second time, the fire alarm somehow mistake some false alarm as a stimuli for your body to fight against it. Then it becomes a disease. That's one of the type, like say, how our neuron or Alzheimer's disease, why this DNA now in this case become harmful to trigger the immune responses and the microglia. It's coming back and it's in totally like a circle, like Mike and Sharon were saying. It's like a circle. It started from beneficial way, and then somehow it was not properly controlled or terminated. It becomes bad in our case. Yeah, that's what this card we are studying, to see how the mitochondrial DNA talk with the immune system to induce all the microglia neuron inflammation in the Alzheimer's disease. Thank you.
Excellent. Thank you very much. If it's all right with everybody, we'll go on through everyone's presentations and save questions for the last, if that's okay. So everybody gets a chance to speak to us. So thank you very much. She did boil 2 hours and 45 minutes down to 10. That's very good. It's very good. So next we have Dr. Unni. Tell us about yourself.
Happy to. So I come to Alzheimer's disease from a slightly different angle. I'm a neurologist. I take care of patients about 20% of the time, and about 80% of my time is doing research. Most of the patients I take care of have Parkinson's disease or Lewy body dementia. So this kind of research started when I was a fellow. I did my neurology training at Mass General Hospital and Brigham and Women's Hospital in Boston. And I worked there with a person named Brad Hyman, who's an Alzheimer's disease researcher. But when I joined his lab, I was interested in Parkinson's disease. So he had developed this approach where you can take a mouse, anesthetize it, do a surgery, and then you remove a flap of the scalp and then a flap of the skull. And then you glue on a piece of glass. And if you do the surgery well, the mouse can live its normal life like that. But you've created a literal window onto the mouse brain. And it allows you to study individual cells in the mouse brain over time, even months at a time. And so he had kind of pioneered that approach to study Alzheimer's disease. Clinically, I was interested in Parkinson's, so I learned that technique and was very interested in studying a protein called alpha-synuclein, which is what aggregates in Lewy bodies. You can sort of think of alpha-synuclein like tau is to neurofibrillary tangles, alpha-synuclein is to Lewy bodies. And so I moved to Oregon about 15 years ago, and soon after I moved there, even though I had spent all this time trying to learn how to image alpha-synuclein in the mouse brain, we couldn't actually create Lewy bodies in the mouse brain. But soon after I moved there, actually, someone you just heard about, John Trojanowski and Virginia Lee, his close collaborator, they developed a new approach that allowed you to create Lewy bodies in the brain, even in a mouse model. I won't go into the details, but I'm happy to talk about later if people have questions. And one of the things we realized by imaging alpha-synuclein in the mouse brain when a Lewy body forms is normally the neuron is full of alpha-synuclein everywhere, including in the nucleus where the DNA is. But when the Lewy body forms, for whatever reason we still don't understand, all the normal synuclein that's in the nucleus disappears. And you have this kind of clump, very much like a neurofibrillary tangle, but not in the nucleus. And through kind of a roundabout set of ideas and of experiments, we kind of stumbled upon this idea that this alpha-synuclein protein is actually really important in repairing a certain kind of DNA damage called double-strand breaks. It helps facilitate that repair. And so DNA is where the information is stored to create a cell or to create an organism. And so if that DNA is damaged, it corrupts that information. And we know in diseases like Alzheimer's disease and Parkinson's disease that there's a high level of DNA damage in the brain. What role it plays in those diseases, I think, is still to be revealed, and many of us here, and I think we're going to hear about later today too, are kind of interested in that idea. But our kind of unique take on it was that this synuclein protein might actually be helping repair DNA. And it struck us that actually other people had published kind of very similar ideas about tau. And it gets even more complicated than that. There's subdomains of the nucleus called like the nucleolus where very specific kinds of DNA repair happen. And our most recent data showed that synuclein is working there. Other people had showed that tau is actually working there too, maybe doing something very similar to what we think is happening with synuclein. So that kind of sparked this new idea in my lab that maybe synuclein and tau might be related to each other in this process. And it could help explain why, even though we define Alzheimer's disease, let's say, by having plaques and tangles, or we define Lewy body dementia by having Lewy bodies, in reality, when you look at the brains of people who suffer from these diseases, many of them have mixed pathology. So Alzheimer's patients, half of them will have Lewy bodies. Lewy body dementia patients, actually the vast majority already have Alzheimer's changes too. So our idea, which I agree is a little bit out there, but it has to do with this idea that DNA damage, synuclein and tau, trying to help repair that, if that process goes wrong, it might trigger these proteins to aggregate. And that might explain why we often see these protein aggregates present in the same brain, tau or synuclein. So I'm happy to talk more about the exact specifics later, but maybe I'll leave it at that level for now. Yeah.
Very good, thank you.
Sorry.
That's all right. And I'm really sorry I didn't try to repeat your question, but there was no way I was going to be able to. This is Dr. Fulin Ma. Tell us about your work.
Sure. Hi, everyone. My name is Fulin Ma. Some of my generation, I'm from mainland China. Some of my generation of Chinese people, parents also give them an English name, just so that folks is easier to pronounce, and I go by Jack as well. So hi. I started my graduate career in 2015 in University of Connecticut. And back then, my major was microbiology. I specifically study foodborne pathogens. A lot of our studies are funded by FDA and USDA. And then later on, when I moved to my PhD program, I want to find something more challenging and makes a bigger impact to our human race. So I followed Dr. Carl Harrop as my PhD mentor in Hong Kong University of Science and Technology in Clearwater Bay in Hong Kong. And I moved to Pittsburgh 2019, right before COVID. So there's a short year-round delay because Pennsylvania shut down during the COVID pandemic. And I officially started my own lab 2 years ago. So my lab works in a more thorough way and a different way to look at Alzheimer's disease. So Alzheimer's disease, as we all know, is a neurodegenerative disease. And neuron or neuro is in the name. But our lab also focuses more on other supporting cells, just like microglia, but specifically it's called oligodendrocyte. So I'm sure you see on Dr. Kapil's demonstration, the cartoon of the axon, and a lot of the axons are wrapped with little bundles. And you might be wondering, what is that? So those are actually called myelin sheath. They're a protoplasm-generated insulation sheath that doesn't come from neurons, but come from the oligodendrocyte. So the myelin sheath from oligodendrocyte is very important in both supportive role and also helps the electric signal to propagate more efficiently. More importantly, the degradation of the myelin sheath, which is the myelin debris, is highly inflammatory. So these myelin debris can be picked up by microglia that Dr. Xian is working on and cause a really catastrophic effect towards neuroinflammation. So my lab have preliminary data to show, and also some of the published data to show that in a Alzheimer's mouse model called R140 mice, these are the type of mice developed by Dr. Bruce Lam from Indiana University that overexpress human amyloid precursor protein with a Swedish mutation. So this mouse overexpressed this human protein, and it starts to show deficit and amyloid plaque deposition around 12 months, which is about middle age. So total lifespan of mouse typically is 24 months. Our lab have found that the death of oligodendrocyte and the loss of the myelin has happened around 6 months, which is almost half a year before the amyloid deposition and massive neuron death. So these are very strong evidence to show that oligodendrocyte damage and death could be one of the cause for the neuronal death that happened afterwards and trigger the whole neuroinflammation effect. So with this CART grant, I'm going to pursue to study the DNA damage in oligodendrocyte that is triggered by amyloid precursor protein. So I'm sure a lot of you folks have heard about amyloid precursor, amyloid plaque and tau tangle. Amyloid plaque come from this precursor protein, as its name indicated. And it's a really massive amount of protein. It's highly expressed in all kinds of tissue, from brain, neuron, microglia, to oligodendrocyte to kidney cells even. As such a massive expressed protein, we're also interested in some of its innate function. And as a matter of fact, we found that oligodendrocyte has higher expression of amyloid precursor protein even than neurons in the brain. And they are actually responsible for about 1/3 of the total plaque load in human mouse model. So to study the effect of amyloid precursor protein in oligodendrocyte DNA damage and death, and the consequence of the myelin sheath loss is very important to study the cognitive function loss in Alzheimer's disease. And that's my research will be for the CART grant support. I would like to take questions after Dr. Calvier's talk. Thank you.
Thank you, Jack. Dr. Laurent Calvier?
Yep, correct. Yeah. So you might have guessed with my name and my accents, I'm coming from France. Some of you I know guessed it already. So yeah, I'm coming from France. I did my PhD, my thesis in France, and unlike most of you, I guess here, I'm coming from a different background. I'm coming from the cardiovascular background. So I did my PhD on hypertension. Then after that, I moved to Germany to do my first postdoc, and I work on a different circulatory system that was the pulmonary circulation, and I work on pulmonary hypertension. And by collaborations, I was very lucky to know my next mentor, who is now in Dallas, Texas, and he was working on atherosclerosis. So this is how I came into the US, and I had to convince my wife to follow me here. I told her initially, you know, I did my first postdoc in Germany. I have this huge opportunity to do my second postdoc in the US with very bright minds. My share in my department, I Nobel Prizes, so it's like a great opportunity for me. Would you follow me for like 2-3 years to do a postdoc, you know? And 9 years, that was 9 years ago. And so yeah, I ended up in a lab being almost the only one not working on the brain. And I was looking at the mechanism allowing the immune cells in the blood circulation to infiltrate tissues. And so my main interest initially was on atherosclerosis. And then we moved slowly, because this mechanism is actually active in different diseases. We were able to block this mechanism to reduce inflammation. And then we were looking into different applications where this mechanism and this drug that we are developing would be the most potent, basically. And so we slowly switched to multiple sclerosis, which also relies a lot on inflammation and the influx of immune cells from their circulation into the central nervous system, the brain, the spinal cord, to degrade the tissue, the myelin there. And so we had also great results here, and we followed a bit more on this pathway, and we ended up testing it in Alzheimer's, and that was something like 5 years ago. So 5 years ago, I knew nothing about the brain. And yeah, now my lab is mostly focused on Alzheimer's, trying to understand how inflammation help the propagation or maybe the initiation of Alzheimer's disease. And applying the mechanism that we found outside of the brain into the brain vasculature and trying to block the infiltration of immune cells into the brain to stop neuroinflammation in the brain. And why this is so important for this specific disease is because once you have neuroinflammation, this creates a bad environment for your cells in the brain, especially the neurons that are stressed by this inflammation and don't function properly. So by trying to reduce neuroinflammation to a normal level, by blocking the entry of these immune cells into the brain, then we can improve the function of the neurons this way. So initially I was not working at all on amyloid. I was not interested into this protein initially because I really was focused on inflammation. And by working step by step into this mechanism, we actually found that by blocking neuroinflammation, you can also reduce plaque accumulation in the brain. And that's one of the active questions that we have in our lab, trying to understand how, by targeting cells in the periphery, you can act also in brain, and especially reducing the plaque and helping the neuron to function better. So that's one of the research axes that we have in our lab. And thanks to this CART grant, now we're going to be able to develop a new axis, which is to find protective mutations to prevent cognitive decline in AD patients. And really this work initiated by two publications already that published case studies about patients who were supposed to have Alzheimer's because they have a gene mutation that would cause Alzheimer's disease. And somehow these patients don't progress toward dementia. They keep normal brain function for decades after they have plaques in the brain. So these were the very two fundamental papers that we want now to follow to try to identify new mutations, new mechanisms that can also prevent the progression toward dementia and preserve the cognitive function in the brain. And that's only because we got this award now, so I'm very thankful.
Well, these folks have done a fantastic job of staying tight on time. So we have lots of times for questions, which is awesome. I'm sure you have some, and I will, if they're not too complicated, repeat them for the streaming audience. Yes, in the back. We're talking about inflammation and proteins. Oh, they're either too functional or not functional enough. Does that, someone want to tackle that? And you can just pass the mic around. This is very informal, so it doesn't have to be.
Yeah. Oh, I can start and then I can pass the mic around. It's really a key question right now, trying to understand the different contribution of different cells into this mechanism. Because initially, your immune cells are able to tackle these kind of issues, and at some point, they reach a time when they are not able to clear it anymore, and this is when you start to have accumulation of this protein. And trying to understand how neuroinflammation promotes the shift of these cells toward from being able to clear this protein to not being able to clear it anymore. Trying to understand what is this shift here is actually an active question in this field. And I'm going to pass the mic around.
Anyone else?
I think it's a great question, and I would say that in a lot of these diseases, we're presented with this issue where there's no doubt that there's inflammation in the brain with Alzheimer's disease. But one of the big questions is that a reaction to something that's happening? And is that a normal thing that the body is trying to do to prevent things from getting worse? Or is the inflammation actually driving the disease? And so, how you would affect the inflammation would be exactly opposite with a treatment in those cases. In the first case, you'd maybe even want to promote inflammation if it's doing something beneficial. But in the second case, you'd actually want to tamp down the inflammation if it's actually driving the disease. And we see this in Alzheimer's disease. We see this in a lot of diseases. And I think the early kind of ideas were that the inflammation is reactive. I think the more we learn, we're starting to realize that actually there are aspects of the inflammation that are harmful, and tamping that down, like you guys have heard already beautifully from several people, is a way that I think people are moving forward. But you can't do it non-selectively. You can't just suppress the entire immune system like we do in some other diseases and get benefits.
I actually have something to say also, because I've been studying inflammation and then now autoimmune disease, or I think Alzheimer's disease is really an autoimmune disease. I think our host or our body is really having the information to deal with whatever stress. That's initially information is designed or engineered to be beneficial to ourselves. I think that's a first concept for information. Information is a way to deal with this stress, to clear out this injury, but it also follows with a resolution. That's a definition of the information. That's also why we have the information from the beginning. But once you have that fire up with these inflammatory responses, so many players, like Laurent was mentioning, so many players already play a role there. You pretty much let them go through this process once. They've experienced something. The cell also have the membrane to learn what they experienced. So they are not the initially whatever they were before the inflammation. That could be this series of this stress which trigger the initial inflammation. Let these cells go through this path. Therefore, these cells are not exactly what they were before inflammation. So your question, I think, is a very excellent question. Also triggered me to, we discussed at the dinner table yesterday, why males versus females, women and men, say infectious disease, pretty much men were not doing as well as women. When you have like SARS-CoV-2 infection during pandemic, we found men are doing worse or easily get sick compared to women. But when it comes to the autoimmune disease, women are doing much worse. Autoimmune disease risk happens more, like Alzheimer's disease also happen more in women compared to men. All the autoimmune disease, like some typical one like lupus or Alzheimer's disease, easily happen in women compared to men. I think there is some genetic difference between the two genders, but this is also an example to tell you inflammation at what stage in infectious disease, what it is for, and why, because research now have shown that in men, actually, our immune responses are weaker than women. Generally saying, some of the responders or sensor to respond to stress is not as active as in females. That could be one of the reasons why when you have the SARS-CoV-2 infection, men are doing not as well as women. But on the other, if you flip aside, on the other hand, in autoimmune disease, actually women or Alzheimer's disease, women are doing worse than men. I think that could be one.
Thank you very much. So now Tiffany has volunteered to be or actually we have the microphone in someone's hand now. So if you have a question, raise your hand, we'll get the microphone to you so everyone can hear it. We have a question on the floor.
Yes, my question is specifically for Dr. Jack Ma. Is that not the coolest name for a movie, y'all, Dr. Jack Ma? Anyway, so given your nutrition background and some of the CART members here, forgive me, I cannot remember the researcher's name, but a few years back we had a researcher and one of his findings that there were 5 foods that decreased inflammation in the brain. It's broccoli, Brussels sprouts, blueberries, walnuts, and whole grains. Coming from your background, have you found that effect on the myelin sheath, just like they have on the inflammation in the brain? How does nutrition affect that?
Okay, thank you. That's a very good question. Just to clarify, my master's training is in foodborne pathogens. It's still microbiology. I did take nutrition in college, though, so don't worry about that. So I think inflammation is big, and as we were talking about it, and if I might add to what Dr. Xian and everyone else is saying, I think inflammation is necessary. You know, it's designed by nature to fight pathogens. But it's really a balance, even in your brain. So our brains are what we call the immune privilege site. That means it has a slightly more mild and different type of immune response when it's attacked by pathogens. There's also other important part, like the human testicles is also another part. Those parts are immune privileged because a very strong immune response can potentially cause more damage than just having the pathogens linger there a little bit and getting wiped out by some innate immune response. So, for example, microglia is a specific type of immune cells in the brain. And I don't think we need to demonize this concept of inflammation, but a lot of times it's really a balance whether to get your brain or other body part colonized by pathogens like bacteria and virus, or having brain inflammation and wipe out those pathogens. So, but a lot of times with Alzheimer's disease, we don't really have those significant amount of pathogens triggering the disease. Instead, it's a lot of the natural breakdown product from aging, from DNA damage of different cell types can generate those debris and what you would call like trash to trigger the immune response. And not to advertise my lab, but we also work with with I'm already there, I guess. So we also work with this cytoplasm DNA in microglia because usually DNA is never supposed to be free roaming in the cytoplasm. It's either in organelles like mitochondria or it's in the nucleus. Having free roaming DNA in the cytoplasm is usually a sign for virus infection. But however, sometimes when mitochondria breaks down or DNA damage, we have found that there's leakage of DNA in the cytoplasm of microglia. And this can cause a fake false alarm for microglia to thought it's being invaded by virus. And this can cause very strong immune response, but it's really targeting nothing. It's targeting its own DNA. So that's another aspect my lab is working on, but it's not really in the card thing, so I didn't bring it out in the beginning. And when it comes to food in general, like I said, for Alzheimer's disease, there's not real pathogens that you need to fight. So a lot of times neuroinflammation is usually a bad thing. So any food that would decrease in general the reactive oxygen species and neuroinflammation would be good. Thank you.
Yeah, if I can add also, that was very comprehensive, thank you. It's also something that Alzheimer's is a multifactorial disease, right? So you cannot only focus on one side, but probably from different approaches you can tackle it. And food is one of these. I mean, you might know that the risk factors for Alzheimer's is obviously aging, but you can also have cardiovascular diseases that can promote the progression of the disease. So if you have obesity, if you have atherosclerosis, that's all going to promote Alzheimer's. The idea that we are following right now is inflammation, obviously. When you start to have inflammation somewhere in your body, all your inflammatory factors are going to go up and all your body is going to respond to it. So you really want to try to slow it down to a normal level. And obviously you have this acute infection, which should be resolved very quickly. That's what we mentioned previously. And if you have like a low grade but ongoing inflammation, this is where it's coming so bad on the long run. And this is where food can be also a way to help against this by having food that decreases the amount of oxidation that you have, lower the inflammation, and basically try to keep the system on check.
Yes, number of us, getting an echo here, a number of us have been watching this process since CART began and found it rewarding. There is one question that comes up to us who work with us, from our people from whom we are raising funds. They want to know how the progress is coming. Every now and then you hear something in the newspaper, a breakthrough is something that really dissipates and you see nothing, maybe an early diagnosis of Alzheimer's, but from what the layman can understand, is it possible for you to tell us how the progress has been coming along in the last 20 years? Now I realize you folks are too young to remember what's been happening since we did this, but can you give us any idea how we've been coming on research in Alzheimer's in the last 30 years?
Yeah, I can start and then pass the mic around. It's really a slow process. That's the true answer. For many reasons, a good one is you don't want to rush to go too fast to humans because there is a lot of control that you want to make sure that you're not going to make it worse, basically. So you're starting with basic research, cells, mice. We had some good examples of what you can do with mice behavior, for example. Try to understand your mechanism there, and then you slowly progress toward understanding, having a drug, trying to test if it's toxic first in animals, different animals, then you progress toward the first clinical trial. And the first thing you do is you give increasing doses of drugs to healthy patients first to see if it's not toxic. Then you start with a small group of patients with the disease, and then you progress. So that's phase 2, and then you progress to phase 3, where you have larger population, 100,000 patients there. And all this process, from the discovery in the lab to having a wide distributed product, takes about 15, sometimes 20 years, especially when you have long-term diseases like Alzheimer's. You cannot just give a drug in clinical trial and come back 2 weeks later to see if it's working or not. You have to have these long clinical studies. So this is this is a challenge for us, really, to try to move quickly to the clinic. And one of the a good thing right now that the research is coming up with is biomarkers, helping to find quicker how effective is your treatment for patient. And you don't have to wait until you see dementia or not. You can already look at biomarkers to reduce the time that you need to follow the patient.
I think it's a great question and one that we get asked a lot. And I think, as Laurent just said, it's not a linear process, unfortunately. It's not like every year you make the same amount of change or improvement towards the goal, of course, which is to find a cure. But it's very non-linear. So if you look at other diseases, as a neurologist, multiple sclerosis is a great example, where it was a devastating disease for many decades. And then there were a few discoveries that really changed things. One was new drugs that could tamp down inflammation. Another was a new imaging method, MRI, that allowed you to have a biomarker for the disease that allowed you to test drugs more quickly. And then when those two things came around, now multiple sclerosis is a completely different disease than it was 20 years ago in terms of our ability to treat it. Now, Alzheimer's, unfortunately, is a little bit harder because it probably starts years or decades before the person actually realizes it. So then the question is, we need to find effective therapies that can help after the disease has probably been going on for a decade or two in the brain. So that just makes it even more complicated. But I think there's no question in the last 20 years we've learned so much about it. And we're all hoping that because of this non-linearity, maybe, we could be on the cusp of something like multiple sclerosis. But it's just so hard to predict, and I get asked that all the time, and I can't really give a satisfying answer.
Just to add something, that's great. That's about the disease. I want to share some personal stories for this CAR-T decades, what advance we can make. So as I was saying, my postdoc mentor was awarded by this 2017 card. That was around 10 years ago. So look at now, I did my postdoc training with him for 6 years, and then now I have my own lab. I become an independent investigator, expanding his work, and then keep working on this field. Hopefully, within the decades or even more time, we can really make some progress, not on just from one lab, from all tons of these scientists, our research, and all the lab people. We can make some progress on the biomarker, on the different diagnosis method, and also the treatment. But personally speaking, I think it's, what's that word, heritage, is it? So it's kind of heritage from my mentor, 2017, he got from CART. And now you look at me, I'm a 2026 CAR-T award in my own lab. So I started my lab 2025, and I was sharing this card for me is tremendously important. I started my lab 2025 fall, September, October in Milwaukee. Then I started to apply for this master cycle after 2 months when I started my lab. Then I got this card within 6 months after I started my lab. So this is really it's a great support it's my first grant I got as an independent investigator. It give me some confidence showing that, hey, you can do this work. And hopefully in 20 years, I can look back, and Sharon told me, you will never forget your first. This is my first grant. And if I get successful in the future, I will always keep CART close to my mind. Yes.
Thank you. So Jack, on the topic of perseverance, I understand you're a fan of Liverpool. Do you have a little metaphor you want to share with us?
Thank you. Yes, I see folks being searching me online. I was the word I put in eNeural editorial panel that I put. I really like Liverpool, and it's it's really a fight. Like just like the logo for Liverpool is "You never walk alone," but it's also about endurance and persistence on a project. So I do agree with Dr. Calvier and Dr. Unni was saying that this is really a long process to develop cure for Alzheimer's. And Alzheimer's is really, I say it's really fundamentally different from almost any other disease because you find the Alzheimer's researcher, us, other people you see, if you want to make them embarrassed or if you want to trigger them, ask them what is the cause for Alzheimer's disease. So 5% of the Alzheimer's disease is genetics. That means there's known genetic form. There's amyloid precursor protein or APP gene, there's presenilin 1 and 2. But that also means 95% of Alzheimer's is sporadic with no known genetic cause. So that's why I see Alzheimer's disease as really like a web and we're like the fish inside. If you want to get out of the web, you don't just clip one link. It's a multifactorial inflammation, oligodendrocyte, lifestyle changes, it all plays in role to help improve the cognitive function and fight Alzheimer's. So it's really important to look more mechanisms. And of the past 30 years, there's a lot of drugs that are targeting specifically on amyloid plaque, lecanemab, donanemab, and you know how those work out. Plaque load are reduced. Some of the cognitive function in the patients are actually worsened. A lot of them are unchanged. So, everyone in neuroscience knows this, and we learn it in textbook. We say it all the time. Dr. Alois Alzheimer found in his patient Auguste that there's amyloid plaque and tau tangle in the brain after she passed from the disease. Back then, it's just called the disease, but Alzheimer named it Alzheimer's disease. And just because you keep finding these two patterns and the debris deposition, whatever you name it, in the brain, sometimes we have to come back and think not necessarily that means those two hallmarks are just the sole cause. So I think as scientists and as a general public, we have to also widen our vision to see what other cause could there be, because it might not really be that one single cause for Alzheimer's. And that's my answer. Thank you.
So, thank you very much. So Tiffany, ask somebody else.
Last one.
Thank you for all you're doing. And what has intrigued me from personal experience is how big a component is genetics in the cause of Alzheimer's?
Okay, so I think I'll take this one for first, and then I'll pass the mic around. So, like I was saying, a very small portion of Alzheimer's is caused by known genetics. However, of those genetic forms, they're very significant effect to the pathogenesis and the ongoing of the disease. Usually, you find it in some families with the risk genes. It's almost inherited that parents and their generation and their kids, they all have some early onset, and that's what we call familial form of Alzheimer's, or FAD. Another big part is the sporadic Alzheimer's disease, or SAD. Folks really focus a lot in science field on the familial form because they're easy to study, because there's so many genes to study. A lot of the mouse models people build are genetic form of the mouse model. So we humanly engineered to put in more of the human risk gene in mouse and to study the familial part of the Alzheimer's disease. But like I was saying, I think everyone also need to think about the other 95%, which is the large portion of the sporadic part of this disease. So to answer your question in the short answer, I think it's very important, but it's only a small part. So yeah, that's my answer.
I will add also another side of this is on the project that I'm proposing here is trying to also see the other side of it. What are the genetical mutations that can protect against Alzheimer's disease, which is going to be obviously very small because not many patients have these mutations, but trying to not only understand what has been done so far, trying to understand what genes are promoting the disease. Now we're trying to look on the other side, what genes are protective against against the disease. So that's also another part of the question that I wanted to bring up.
Yeah, I would say it's a great question with a complicated answer, because genetics itself is very complicated. And as Jack was just saying, 5% of cases are genetic in a simple sense, where we know you inherited one bad gene from one of your parents. And those genes are so powerful in terms of causing Alzheimer's that just having one bad copy causes the disease. But, there's probably about roughly 20,000 genes in the human genome. And in the 95% of cases that are sporadic, we don't know what causes it. But some people do think that in those cases, maybe genetics still plays a role. It's not like a simple one gene inherited from one parent causes the disease, but maybe if you inherit a few different genes from both your parents, each one by itself maybe doesn't cause the disease. But if you're unlucky enough to get that combination of 5 or 6 genes that each have a small effect, and then maybe you throw on environmental interactions that might also predispose you to Alzheimer's disease, then that's the combination that gives that person the disease. So this is a hypothesis. We don't have strong evidence yet that that's exactly what's happening, but it's one that many people, and I myself, think it's very reasonable, because there are other diseases like hypertension and high cholesterol. We know many people who have it. There is some genetic component in most of those people, but it's not like a simple one gene. It's a great question, but a complicated answer.
I just can add a little bit. I think genetics risk is most important and prominent one for majority of the disease. Doesn't matter, cancer, Alzheimer's disease, or vascular disease. But we do have some environmental risks also. When you have, for example, I was using the COVID all the time as an example. We have long COVID now. And then the long COVID, after the viral infection, a couple of years later, all kinds of symptoms came up. Now it popped up with even the neuron dysfunction. So I would say genetic risks are really important and very permanent to study, but we should not also ignore there are some non-genetic risks, like infections. Like we are keep talking about the inflammation, then after it's resolved, the second wave, how they come from. It could just come from some infection or parasites infections. Yeah, it's still possible, and then we should not ignore that part also.
Excellent. Well, the beauty of this event is that you have access to these brilliant minds over lunch and for the rest of the day. They're very accessible. Many of you had dinner with them last night. That's really the fantastic part of this event that is unparalleled, and it makes it very special. So I want to thank all of you for your contributions and your comments, and it was just a pleasure meeting all of you.